Good evening, everyone. My name is Talia Brown, a student at the University of Technology, Jamaica, studying the Real Estate Management and Valuations course. Today, I will be sharing my views on the social significance of gene editing technologies, like CRISPR, which has ignited optimism alongside ethical concerns all over the world. Let us delve into how CRISPR is transforming our world. So, what is gene editing? According to Medline Plus, gene editing is also known as genome editing, which is a group of powerful technologies that give scientists the ability to change an organism's DNA. These technologies allow genetic material to be added, removed, or altered at particular locations in the genome or the gene. A well-known one is called CRISPR-Cas9, which is short for clustered, regularly interspaced, short palindromic repeats, and CRISPR-associated protein 9. According to Medline Plus, CRISPR arrays in bacteria function as a memory system, enabling them to recognize and disable viruses. Researchers have adapted this system to edit DNA by creating a guide RNA that attaches to Cas9 enzyme, directing it to cut DNA at specific locations, similar to how it functions in bacteria. It's like a molecular pair of scissors that can cut and replace specific genes with unprecedented accuracy. One of the most promising applications of CRISPR is in medicine. It is being explored in research and clinical trials for a wide variety of diseases, including single gene disorders such as cystic fibrosis, hemophilia, and sickle cell diseases. It also holds immense promise for the treatment and prevention of more complex diseases such as cancer, heart disease, mental illness, and HIV infection, according to Ernov F. 2022 from the New York Times. For example, in a groundbreaking study published in Nature Medicine in 2020, scientists successfully used CRISPR to treat a patient with sickle cell disease by editing their stem cells. Even though there are successes, there are also tragic failures in clinical settings, as per Linosi et al. 2018. Apart from its medical applications, CRISPR also influences agriculture and environmental preservation. As per Linosi et al. 2018, researchers are investigating its potential to develop crops with increased resistance to pests and diseases, which could lessen reliance on damaging pesticides. Nonetheless, worries persist regarding the environmental consequences of genetically altered organisms and the potential decline in biodiversity. However, in addition to editing somatic cells, which are cells that make up most of the body, it's possible to edit genomes of gametes, which are eggs and sperm, and early embryos, called germline editing. Any such edits in humans would not only affect an individual, but also his or her progeny. They could also theoretically be used to enhance desirable traits instead of curing diseases, as per the Jackson Laboratory. Critics argue that these possibilities raise profound concerns, as now you have the issue of the possibility of creating designer babies, where parents could select desirable traits for their children, leading to genetic inequality and societal divisions. Just imagine the gap in society and the social implications that could cause, according to Medline Plus. Another concern is the consequences that gene editing may bring in the long run, in which we may not know or understand now. Some may also argue that equality and availability are crucial elements of the CRISPR breakthrough. Despite its potential benefits, there's a concern it could make existing healthcare gaps worse. Who gets CRISPR treatments? Will they be cheap or open to everyone? Or will they make the divide between rich and poor wider? While concerns about designer babies are valid, stringent regulations globally prohibit the clinical application of human germline genome editing. According to the National Library of Medicine 2022, the clinical application of human germline genome editing is prohibited worldwide. Basic research in this field is also subject to rigorous oversight. These measures ensure the CRISPR gene editing is ethically vetted. Ongoing research aims to enhance the safety and efficacy of CRISPR techniques, alleviating concerns about unintended consequences, as per Zimmer C. 2022 from the New York Times. Thus, existing regulations and ongoing research effort mitigates risk, fostering responsible CRISPR technology use. To conclude, CRISPR gene editing represents a groundbreaking advancement with far-reaching implications for medicine, agriculture, and environmental conservation. While the technology holds immense promise for addressing genetic diseases and improving crop resilience, it also raises significant ethical and societal concerns, particularly regarding the potential for designer babies and exacerbating healthcare disparities. However, existing regulations and ongoing research efforts demonstrate a commitment to responsible governance and continued innovation in CRISPR technology. Thank you for listening, and I would love to hear your views on the social significance of gene editing technologies such as CRISPR.